Jeff and I started talking about biotechnology, and perhaps not surprisingly, we decided to talk about genetically modified crops. So we put up the learning objectives again, so we all remember where we're going with this. So what are genetically modified organisms? We should be able to compare different methods, breeding, mutagenesis, and GM. So we talked last week about both breeding and mutagenesis, and I'll finish the actual gene transfer today. And then we'll talk about the costs and benefits of genetic modification technology. And of course, you've seen that we're trying to integrate things from throughout the quarter into this section. Oops, wrong button.
It's just
Wrong button. Okay. So we started off by talking about artificial selection or breeding, which is the process by which heritable traits become more or less common in a population based on the desirability of those traits to humans, which is of course a little bit different than natural selection, where it's the natural environment sort of putting selective pressures on different organisms in the environment. In this case, it's humans putting the selective pressure on the different crops, and you can see that people selected for plants that are shaped in a way that are much easier to grow in an agricultural field. And then also people were selecting for food sources. So instead of teosinte, which are those hard, tiny, only a few kernels, more like modern corn, which are big and plump and juicy. So that was artificial selection. The problem with artificial selection, of course, is that you're limited by the variation that's actually found in your population or in the population of the farmers near you, limited by what's available in the species. And I know this is sort of a silly example with wanting to breed a dog with wings, but I think it makes the point really well that if the variation's not there, you can't breed for it with traditional methods. And so that brought up some of the ways that people now modify the genomes to actually introduce new traits. And so one of those was breeding by mutagenesis. So this is an example of using radiation to make changes in the genome. And you can see that this radiation made some changes in the genome, and now we have these beautiful light blue flowers in addition to those purple ones. Also, you can use a chemical called EMS to introduce mutations in the genome, and that's where ruby red grapefruits that you've all seen and probably tasted come from, is through random mutagenesis using chemicals. Introduce a change in the genome, acquire this new trait of ruby red grapefruits. And so that's great, but that is random. And you're still limited by what was already present in the genome that you can actually change or alter with just random mutations. So then we wanted to talk about actually taking genes from one species and putting them into another. And so that's sort of where we're going with this genetic modification technology. So everything I've talked about up until now is not considered genetically modified in the sort of legal definition. So breeding, random mutagenesis, that is all not GM. But now taking genes from one species and putting them into another is now this genetic modification. Jeff talked briefly about the gene gun, and then I'll finish talking about this agrobacterium-mediated gene transfer. So if you remember the gene gun, you can take DNA or the sequence of the gene that you want to put into a new crop, attach it to gold particles, put your gold particles with the DNA into this machine, and shoot those gold particles at your plant tissue, and with some frequency that DNA gets incorporated into the genome of the plant tissue. This is, of course, sort of a messy process because you're just shooting DNA at your plant cells, and so the insertions are not always nice and clean. They can be sort of messy, and so that's one big downside. The other big downside that we talked about is that you can't target where the piece of DNA integrates into the genome, so it could land anywhere within the genome. And then, of course, we know that only certain cells actually go through meiosis to make gametes, and only the gametes go on to make the next generation. So transforming leaf tissue doesn't really do much good as far as developing a new trait in a crop to be passed along to the next generation. So people shoot those gold particles at this undifferentiated tissue, and then once that tissue has been transformed, then through these tissue culture methods you can regrow plants that are transgenic. So there are questions about that stuff from last week that Jeff talked about before I go on. Okay. So the next thing where I picked up was with this agrobacterium-mediated gene transfer. So if you remember, agrobacterium is a bacteria that is commonly found in the soil. It's able to recognize wounded plants and actually then transfer part of its genome into the plant and integrate that part of its genome into the plant genome. So here's the agrobacterium here. So let's take a closer look at the agrobacterium. So the bacteria, it has this circular genome here. It also has this additional piece of DNA called a plasmid, which is a circular extra chromosomal piece of DNA. And it is part of this plasmid that actually gets transformed into the plant. So taking a closer look at this extra chromosomal DNA, the plasmid, we have this region here, the tDNA, that gets transformed into the plant, and then the rest of the plasmid that is there to help get that piece of DNA out of the bacteria into the plant and into the genome. So what we really want to focus on here is this area between the left and the right borders, because that's actually what's going into the plant. Oops. Okay, so here we're now zooming in on just this part, since just